And these natural antibiotics are very, very important because we all know that the current antibiotics what we have are becoming resistant quite quickly. Antibiotic resistant bacterial pathogens, so that they list all of the pathogens depending upon the priority of them with respect to the resistance developing against the antibiotics to treat this infection, and why it is important that we need to focus on them. So they usually publish every year the priority pathogen list. So the 2024 list of priority pathogen is given here. And you can see on this diagram that the pathogen list has got 3 groups, which is the critical group, high group, and a medium group. And the critical group has very important bacteria. They are Acinetobacter baumannii, Enterobacterales, and carbapenemase resistant Enterobacteriaceae as well. So this is a very critical group because whatever advanced antibiotics we have, the resistance is developing against them in this critical group of pathogens. And they all are gram-negative bacteria. And then there is a high group, which could be as a medium group of priority pathogens. And they are Salmonella typhi, most of the species of Shigella, Enterococcus faecium, Pseudomonas aeruginosa, another important pathogen to look for. Then the non-typhoid Salmonella, Neisseria gonorrhea, and Staphylococcus aureus. And then comes the medium group. So all the streptococci, Streptococcus pneumoniae, Haemophilus influenza, and group B streptococci are part of the medium group. So all these pathogens are indicated by WHO in 2024 as a priority pathogen, and they are showing resistance to antibiotics which are available right now in the market. Last year, another group was added, which was Mycobacterium tuberculosis, as the rifampicin resistant in these cases are on high level. And especially in Asian subcontinent, I would say, the resistance to TB drugs is really on high, and it is regarded as a silent pandemic because of the cases. So the surveillance report published by WHO related to the global antibiotic resistance says that 1 in 6 bacterial infections globally has now resistance to antibiotics, which is very worrying because 1 in 3 of urinary tract infections are now resistant to any of the antibiotics available. And this resistance is actually to the life-saving drugs such as the fluoroquinolones and the 3rd generation cephalosporins. And this problem is particularly more into low and middle income countries where the diagnostic and surveillance capacity is still remain limited. So the report has data from about 104 countries, and it shows that the resistance is increasing mostly among the gram-negative pathogens like E. coli, Klebsiella pneumoniae, and Acinetobacter species. And what it is doing is it is causing real threat to the effectiveness of our key watch antibiotics. These are the important antibiotics what we have. And the countries with the weakest health system, it faces the biggest burden of this threat. So that is why it is called as a syndemic of resistance, inequity, and limited access to effective treatment. So the word syndemic means you already have one set of problem in the face of all this resistance to the antibiotics. But in those places, you also have inequity when it comes to the access to the healthcare system. And that is causing additional care issues. And that's why it is very, very important that we address this. So for gram-negative pathogens, there is actually urgent need to have a new therapeutic drugs available. And why this is becoming really worrying, because of the carbapenem resistance enterobacteriaceae, which is CPE, and you might have heard them. So carbapenem, these are our main antibiotics available to treat the current antibiotic resistant bacteria. But then Klebsiella pneumoniae, E. coli, and Pseudomonas aeruginosa have shown that the carbapenem available cannot be treated this infection. And this is really worrying, especially for Pseudomonas aeruginosa, because the chronic infection of that is associated with cystic fibrosis. And cystic fibrosis patients, if they get Pseudomonas aeruginosa infection, it increases their mortality rate. So the infections are frequently cannot be cleared by tobramycin or azithromycin. And then you can imagine how worrying it can be for somebody who's living with cystic fibrosis if the important drugs are not working, and then the multidrug resistant Pseudomonas aeruginosa comes into picture. So cystic fibrosis is a disease of lung, where the lungs are in, and if those lungs are infected with Pseudomonas aeruginosa, then the mortality is quite high. So what has happened over the past is the first antibiotics was discovered about 100 years now, like 1928 penicillin was discovered. And over the time, we got new antibiotics, but in last more than 25 years, I would say, there is a proper discovery void for antibiotic research or antibiotic discovery, that we do not have any antibiotics after 1987 discovered for clinical use. And this is very worrying because on one side, we have antibiotic resistance increasing, and on the other side, the discovery void has happened. So we do not have proper drugs available to treat this now. So that is why the resistance is a major global threat now. So what we'll have to do is find alternative strategy for the discovery of effective antibiotics. And to do that, Walker Lab was exploiting the potent narrow spectrum antibiotics, which is produced by many bacteria for intraspecies competition. So what happens is, in any place when bacteria are growing, when there are different types of bacterial species, inherently some bacteria have a capacity that they kill other bacteria, which is called as an intraspecies competition. So to kill that bacteria itself produce a bacteriocin, and bacteriocin is actually a natural protein antibiotic, which is produced by bacteria. So if we could use this natural bacteriocin, that can be developed into new antibiotics. The important characteristic of these bacteriocins are, these are potent, that means it is used in very, very low concentration and it is still very effective. And it has got targeted action, which means that you do not have to worry too much about the side effects on the natural flora, because this is very targeted bactericidal activity. So this type of bacteriocins could be developed into clinically useful antibiotics against high drug resistant gram-negative pathogens, which there are very few therapeutic options. So that is how that was the foundation of the project I was working on. So protein antibiotics, which are the bacteriocins, as I said, this is a potent entity, and it is produced by gram-negative bacteria. They are usually about 40 to 70 kilodalton. E. coli produces bacteriocin called as a cholicins. Pseudomonas aeruginosa, they produce bacteriocins and they are called pyocins. And Klebsiella produces klebicins. And the mechanism of action of these bacteriocins are varied. Some of them kills the other bacteria by DNase activity or by RNase activity or by forming pores or inhibiting cell wall synthesis. On the right hand side, you could see this is a bacterial cell wall. This is outer membrane and this is inner membrane. And as you know, gram-negative cell wall is very, very difficult to break into. What it does, bacteriocin, colicin is one type of bacteriocin in E. coli. It actually enters into the bacterial cell by a cytotoxic domain, and it breaks into it and forms a pore and enters into it and kills the bacterial cell. So it has evolved efficiently to kill specific bacterial cells. So this is most potent antibiotic against the gram-negative bacteria when it was discovered. So the multi-domain protein antibiotics in Pseudomonas aeruginosa are called as S type of piosins, cledicins in Klebsiella and colicins in E. coli, and they have various mechanisms to go into the outer membrane of gram-negative bacteria and then work as bactericidal agent. So the piosins of Pseudomonas aeruginosa, they target several different outer membrane receptors. These are involved into uptake of the essential nutrients like iron. And piosin L1, it has also shown that it can bind to the CPA, which is a common polysaccharide antigen, and which is then a major surface antigen produced by Pseudomonas aeruginosa. And this is mainly important for cystic fibrosis patient because that has shown good activity in that. So the cellular targets of bacteriocin is highly conserved, which is also very important because then you know what to select for. With the cytotoxic activity most commonly taking the form of nuclease activity, it can target the DNA, ribosomal RNA, transfer RNA, or it can just create pores into the membrane. So going into more details of this, the focus is on Pseudomonas aeruginosa and the bacteriocins produced by Pseudomonas aeruginosa, and they are called piosins. And there are few types of piosins. So the piosins S1, S2, S3, and AP41, they carry out their activity by disintegrating DNA, and that's why it has got the DNase activity. Piosin S4 kills the transfer RNA. S5 causes pore-forming toxin, so it creates the pores and then it disintegrates the cell membrane. S6 is carrying out the activity by ribosomal RNAs, and piosin M, it degrades lipid 2,1. So we selected a few of these piosins, piosin S2, S5, and L1 and AP41, and checked how do they behave into murine lung. So these are the mice lung tissues, and the activity was checked on them. So the spot test, which you can see under A, is carried out by having a plate on which Pseudomonas aeruginosa is already growing. So this is Pseudomonas plate. And then you treat that with the piosins, is S2, S5, L1, and AP41, and you can see the clear zone of inhibition. And AP41 is very potent, so it's completely clear. So the presence of clear zones indicates the piosin activity in the lung tissue. And then this in B, you can see it is a staining of the section of the treated lung, so the lung which is treated with the piosins. And you can see there is lack of inflammation or neutrophil influx under piosin S2, S5, L1, and AP41 compared to the control group. So piosins are stable and they do not cause inflammation or tissue damage. So even if they are potent, they are safe for the tissues. So the next experiment, the mice were given piosin 75 microgram, and then the bacteria were counted to see how many bacteria can we recover. So what happened into the first one is the mice were treated with piosin 1 hour post-infection. That means you'll have mice. You will infect the mice with Pseudomonas aeruginosa. That means you're causing the bacterial infection in mice first. And after 1 hour of infection, you treat that mice with pyocin. And then in A, the mice were killed after 4.5 hours. And in the second one, the activity was checked for 24 hours. So in the first one, you can see even after 4.5 hours of the treatment, the pyocin S2, L1, S5, and AP41 have a very good activity compared to the control ones. And among all these four, S5 is very effective, that the cell count is quite low in this group. And after 24 hours, we do not recover any of the Pseudomonas aeruginosa from S5 treated mice lung. So it shows that it has very good activity, especially S5. Then what we have done was we checked the tolerance of pyocin and whether that can be overcome by treating with range of pyocins. So this is very important because we know that the resistance develop quite quickly. So any antibiotic, a new antibiotic, it might take 3 to 4 years, and there is already a resistance to that antibiotic. So if that happens, then what should be the strategy? So the pyocins, if they can be treated, if they can be available as a combination, then the resistance will not occur as quickly. So what we tried here is activity of pyocins S5, AP41, and L1 was tested against the wild type Pseudomonas strain, P8, then a strain called P8AP41T, which is a tolerant strain for AP41, and then AP41 star, which is recovered from the untreated control of mice. And what we then do is you add 200 microgram of the purified protein, spot onto the bacteria growing plate, and check the activity. And you can see, again, you'll have the zone of inhibitions very nicely displayed here. When you count the bacteria, again, compared to the control group, the bacterial count is quite low. And we shows that here in C, that the P8 infected mice, which is a Pseudomonas aeruginosa wild type strain, after 1 hour post-infection with pyocin combinations, they survive and the bacterial count is quite low. So this is very important to see that the acquired tolerance to pyocin can be overcome by treating with range of pyocins. Then a comparison was carried out between pyocin S5 and tobramycin. The tobramycin is important drug which is used in cystic fibrosis patients, especially for their lung infection. And you can see the concentration of the drug. So tobramycin was used at 75 nanogram, and S5 was used as 750 picogram and 7.5 picogram. And at such a low concentration of S5, there was a very good activity on mice treated with after 1 hour of post-infection. So another thing then we wanted to check was, can pyocin S5 afford the protection against Pseudomonas infection when pyocin S5 antibodies are present? And this can be seen here, that the bacterial counts again were checked after carrying out the experiment where multiple doses of pyocin was given intranasally to the mice, 2 weeks apart over 4 weeks intranasally. That's important. At 13 weeks, mice were infected. So first the drug was given, and then the mice was infected with Pseudomonas aeruginosa wild type strain, and then treated with pyocin S5. And then you can see the level of protection given by that group, and then the presence of antibodies. So you already have given S5, and then the IgG count is there. Then after the infection, you see that the protection is still there. So that's why you'll see that no biosyn S5 specific IgG was detected in any of the animal tested. So the conclusion of this study was Pseudomonas aeruginosa specific bacteriocins, known as biosins. They show strong efficacy in a murine model of Pseudomonas lung infection, with the concentration of biosyn S5 required to afford protection from lethal infection at least 100 times lower than the most commonly used inhaled antibiotic called tobramycin. And they are stable in lung. They are poorly immunogenic at high concentration, and the efficacy is maintained in the presence of biosyn specific antibodies after repeated biosyn administration. So bacteriocin encoding genes are frequently found in microbial genomes and could therefore offer a ready supply of highly targeted and potent antibiotics active against problematic gram-negative pathogens. So these were the conclusions, and this was the research carried out to develop this antibiotic further. Because S5 has constantly shown that the high efficacy against diverse isolates of Pseudomonas aeruginosa. They are about 80 to 89% of the Pseudomonas aeruginosa isolates shown activity with S5. And the important thing, or it's very critical, that it is active only against the target organism, so you do not get any collateral damage. And the biosins and colicins are generally stable. They are monomeric, monodispersed, and can be lyophilized. So what is important for any natural antibiotic, any protein antibiotic, to develop as antibiotic for treatment is like, can this be converted into an active drug? What is its drugability? And that was tested in this project, where the primary objective was to generate the methods for the production and formulation of biosyn S5 and a safety profile for delivery by nebulization to the lung. So first, you have to develop a GMP process for the purification and characterization of untagged biosyn S5. Then you have to identify compatible excipients and a suitable commercially available delivery system. Then you have to characterize the long-term stability of the formulated protein. And then you have to perform preclinical toxicology and safety pharmacology studies. So all of this was proposed and carried out as a part of this grant to develop this new protein antibiotic. And I'm not going to show any of the data obtained during that period, but I just want to grab your attention to this slide. It might look a little busy, but I'll talk you through to show that what are the tasks and milestones when you're developing a new antibiotic. So the first is module 1, and it is done into the university lab, where you express the protein and you purify the protein and do the various purification studies. That what is the method for isolating that protein, what is the method for purification that protein, and how effective that is. Once protein is purified, then it has to go into the formulation development and stability studies. And that stability study has to be on a pilot scale, which can then go into the large scale into the industry. So these two things were carried out into the lab. And once that was completed, it was handed over to contract manufacturer. So you would see that the process development and scale up was then transferred to the CMO. Then I'm just trying to find my arrow. Module 4 is CRO1, and you can see into this one that the job of the contract manufacturer was to have the to find a GMP manufacturer where they could have the regulatory approval for their formulation. and then that can go into the first phase of clinical trials. That was for formulation. Another important thing was to find out the stability, so the toxicity, toxicological study of the drug. And this was carried out by a contract research organization. So it's a multidisciplinary project where then you send your formulation to this research organization where they will be able to do all the pharmacodynamics and pharmacokinetic studies for you. And they would guide for the repeat dose related to the toxicity. At the same time, you need to know the long-term and short-term stability of the drug. And that one was the short-term stability was carried out in the university lab. The long-term stability was carried out on the university lab as well on a pilot scale. And then that was handed over to the CMO to see at the large scale what happens. So another important part of this process is the consultation from various experts. And those various experts are from the clinicians. That means those people who are working in the hospital and actually treating the patients. So what doses are working, what type of delivery system they prefer, and what type of patients they have. Like cystic fibrosis is affecting young children quite a lot. So the delivery system has to be worked well for them. And that consultation has to happen between the clinicians and the manufacturers. And then there is expert regulatory input by MHRA, and that's the regulatory body in the UK. And once all of the data is gathered together, then this is handed over to the grant approval body. And then further grants were applied, where this was then taken into spin-out company. So the main conclusions from the study were to actually see the in vivo activity for efficacy of the Piacin S5. So in this slide, you would see that the survival and significant reduction in bacterial load in the acute mouse model of lung infection with Pseudomonas aeruginosa with the strain P8, which is a wild type strain, at the concentration of 300 nanogram per mil, that is 0.025 mil dose. And we have checked the long-term storage. So when you have the drug which is stored at various time intervals, you take it and then you try to put it into your infection model and see its activity. And whether the activity is still there, that will determine its efficacy. So I won't go too much into the detail of this, but what we found is that Piacin S5 is really efficacious in treating the lung infection. So the conclusion of the project then was the research cell bank was prepared and it was confirmed for absence for phage infection and then sent to the contract manufacturer organization. Then the protocol for Piacin S5 expression and purification was developed and handed over to CMO. And then the short-term and long-term stability was determined for Piacin S5. And then Piacin S5 was produced at the CMO as per the requirement. So in this process, as you can see, there were two main branches of this. One is actual checking the physical characteristic of the drug and bringing it to that stage, and then checking the in vivo activity of the drug at the doses at which you are checking your stability. So little bit more information I would like to share about the process of expression of protein and purification. So this was carried out by ion exchange chromatography, where the protein was first expressed into the bacterial cell. You grow it at certain concentration. You harvest your bacterial cell culture. You then isolate your protein from the culture, and then you subject that to the chromatography. And it was done by cation and anion exchange chromatography. And then purified protein was then tested on gel to see whether it is, how pure it is after first ion exchange and then after the second ion exchange run of the chromatography. As you know, I don't know how familiar you are with protein expression. You usually have a tag to the protein so that it can attach to your column. But then it is important that that tag do not affect the activity of your drug. So then you have to remove the tag as well, which you have used for purification. So when you remove the tag, the activity is better and there is less toxicity. So protein purification is actually a very critical step into this project. And once you have that, you have to prove that it can be done at large scale. So for the pilot scale, when you show that maybe you are getting 5 milligram of protein by having your 3-liter culture, then when the contract manufacturers have to scale it up, what level they can do it and how efficiently they can recover the protein. So that was carried out, and I have not included all of the data because this is part of the project, which has to be submitted and has to remain confidential. But we were very successful into getting that that protein Piocin S5 produced at the level where it can be handed over for large-scale production.